Hey, it's Peter here with my FSHD. Well, we're back. You probably didn't expect to hear from us so soon, or maybe you did because you know what's coming. The clarification podcast. Actually, the reality is there's been a lot of action, or news, I should say, in the clinical trial space. A lot of updates have been put out that I wanted to get into. And also I want to expand on some of the topics that Brad brought up. When I do the back and forth with Brad, we don't have a plan. You might, I'm sure you're shocked, but we don't have a plan ahead of time. He just fires away his questions and you kind of hear me going off the cuff and starting to understand, right, why no one else has a podcast. Because no one else is dumb enough to do it, put themselves out there, record it, and then post it online. But I am that dumb. Okay? You know that. All right, so let's get started. Okay? So let's just start with the, you know what? Actually, I want to start with the big thing that's been weighing on my mind. And that is, one of the questions that came up was, which trial would I join if I were an FSHD patient? And that's kind of, I've always said it's very difficult for me to put myself in the position of any one of you. It's very easy to sit here without FSHD and say, oh, wait for this or wait for that. Well, functionality, is not going away. Whereas the clock is ticking, time is muscle. But again, everybody's in a different spot and everybody has a different level or tolerance for risk. But I've got to think of it for a little bit. At the time, I said, I'd join the Avidity. And I've actually come to rethink that. Okay. And I will just tell you right off the bat that right now, today, what is this? March 26th, 2026. With what I know today, if I could join any trial that is currently going right now, I would actually join the Sarepta Arrowhead siRNA trial. I'm not sure what it's called. It used to be called the ARO-DUX4 when Arrowhead was, at least their FSHD project was acquired by Sarepta. It's an siRNA. They haven't released any actual DUX4 data yet, but they have released some preliminary findings. Conceptually, the siRNA approach is this similar to Avidity, but I believe that they're actually, at least the early indications are, that it is overcoming some of the barriers that Avidity has found, or some of the roadblocks that Avidity has found. And we'll get into that more later. But I just wanted to give you right off the bat, because it actually has been weighing on my mind since I said that. I was thinking, and so that's one of the things we're going to do, actually. I'm going to go through, we're going to talk about the Roche news. We're going to talk about, there was some EpiCRISPR news. What else did we have up there? There was, you got the Sarepta news. Going to go through some of the clinicaltrial.gov website and talk about some of the other trials that are ongoing. And so we'll get into some, get back a little bit more sciencey now. Okay. And all right, I'm gonna share my screen right here. All right. So a couple days ago, Roche announced that they are stopping their maneuver, whatever, clinical trial. Okay. And this was their number of industry things where this came out, and they're stopping. This is a myostatin inhibition trial. It's a monoclonal antibody against myostatin. And they were in trial for FSHD and SMA. And we'll get into the idea again. But this is basically the letter from Roche. And again, we appreciate that all of these companies get into the FSHD space. It's a tremendous effort and a tremendous cost to run a clinical trial. So nobody gets into this lightly. And it's a disappointment for everybody when something fails. But there are, though, some trials that have a better likelihood for success and a less likelihood for success based on what we know. And my perspective, as we voiced earlier on previous podcasts, was that this never actually had much chance of success, mainly because this approach has failed multiple times in FSHD. Now, I always tell you, the devil's in the details. There are many different ways to impact the myostatin pathway. We'll talk about that in a second. But this is basically what Roche said. Their trial, the GYM 329, their antibody, it's also what's called mgrobart. And the maneuver trial, it's a phase 2. It was safe. Okay, so safety, and that's always important. But basically, as they say here, it did not consistently deliver the hoped for improvements in muscle growth and function in people living with FSHD. Okay, no problem with safety. So if you're on it, that's not a concern. It just wasn't showing the efficacy that they were hoping for. Now, again, phase 1-2 is about safety and tolerability, but there are always secondary outcome measures, and to kind of tell you, I mean, just because something's safe, you don't necessarily want to go to phase 3, unless you're fulcrum. But, honest. But, no, in my opinion, it's going to be very safe long term. But, you do have these secondary outcome measures on functionality and strength, and theirs did not give them a good business reason or a good science reason to go forward. Okay, so what do we talk about myostatin? So everybody gets excited about myostatin because you basically have seen pictures like this. These giant cows, or giant dogs that have mutations in myostatin, okay, and things such as that. And so what is this? So myostatin, MSTN, okay, is a negative regulator of muscle growth. So, right, when you express myostatin, it binds to its receptors. There's a latent form of myostatin that gets processed, then the active form binds to receptors, and it starts a signaling cascade that leads to a decrease in protein synthesis, increase in protein degradation, and overall can lead to muscle loss. But essentially, it maintains your muscles about the size that they are. Okay, now, when you exercise, myostatin levels go down naturally. When myostatin levels go down, your muscles can actually then start building. Protein synthesis goes up, protein degradation goes down, and you can actually put on muscle mass. That's why it's important. You exercise and you can build muscle. When you have injury, myostatin stimulate regeneration naturally. Myostatin levels go down. Okay, you've released this inhibition on growing muscle. So the idea here is to artificially block myostatin, and if you artificially block myostatin signaling or activity or binding to a receptor or any what you jiggle, that you will then release the hold on building muscle, and you can now put on muscle mass. And in FSHD, where a lot of your muscles are pretty healthy and you have a lot of your muscle cells are not expressing DUX4, the idea would be maybe you could actually bail out the boat faster and put on muscle mass without stopping the destruction. Okay. Another way to do that is to increase follistatin. Follistatin is a negative regulator of myostatin. So actually, if you increase levels of follistatin, you can actually block myostatin, and you get the same effect. High levels of follistatin, low levels of myostatin. Low levels of myostatin, you're allowed to basically have muscle regeneration. Okay, that's kind of the general idea. So you can do it one of two ways. Directly decrease myostatin, that's what Roche was trying to do. Or you can increase follistatin. There's AAV follistatin. There's experiments, and people actually go to Mexico and get injected with follistatin peptides. Bodybuilders do these things. Again, now the thing to remember, okay, in these things is, getting back to our cow, these things, when you see these massive inductions of muscle mass and sometimes strength that goes with it, these are generally healthy conditions, okay? And so when you are basically not undergoing regeneration, you have high levels of myostatin. Blocking myostatin, you can then put on more muscle mass, and that's what's kind of going on with the cow here. Now, when you have muscular dystrophy, your levels of myostatin are actually quite low, okay? And so I'll actually, I'm going to pause right here and I'm going to show you something. Okay, I just pulled up this figure here from our FlexDux4 mouse paper. Now, the FlexDux4 mouse is the most commonly used FSHD model for therapeutic interventions in the FSHD space. We made it in our lab. Dr. Go Jones, my wife, made the mouse, and it's a tremendous tool for studying FSHD therapeutics. So what I'm showing you here are the levels of myostatin mRNA in the mouse. So natural levels, normal levels are arbitrarily here. Let's say it's 1.2. When we put in the DUX4 gene, we get very little amount of transcription. The levels go down. In the uninduced model, the mild model, this is our standard FSHD model. Okay, you're getting regeneration, DUX4 expression, very little DUX4 expression, but enough DUX4 expression that the muscles are starting to deteriorate and you get a phenotype. You can see myostatin levels are going down. In the moderate model, this is a tamoxifen-inducible model. When we induce even more DUX4 expression and more pathology, levels go further down. And in the severe model, where you have lots of FSHD pathology, a lot of DUX4 expression, there's almost no myostatin levels. So now think about this. The bull that we saw, or when you talk about bodybuilders or people at the gym that don't have muscular dystrophy, their myostatin levels are up here. And now if you're going to inhibit that, you can actually then, so you're blocking regeneration up here. If you can release the lock on muscle regeneration, you can then put on lots of muscle mass. Okay, you're going to bring it, you want to bring these levels down. But if you have muscular dystrophy, your levels are down here. Your myostatin levels are very, very low because you're actually undergoing muscle regeneration. You're always regenerating. This is why you need more protein in your diet and things like that, because your muscles are regenerating. Now, you might have some very healthy muscles like we showed in the previous podcast. You might have some very diseased muscles, anything in between. But in general, the myostatin levels are low. So now you're trying to decrease something that's already very low. So you've already essentially released the lock on muscle regeneration, okay, and you're just trying to release it even further. Now, this is mRNA. Myostatin is a protein. Myostatin protein levels do exist in a latent form, so protein levels don't completely track with RNA levels, but it depends on your therapeutic. If your therapeutic is an antisense RNA, for example, this actually matters. Roche was an antibody targeting the protein, so it's a little bit different, but it's the same general idea. You know, you're already regenerating, so you're trying to boost more regeneration in a system that is already regenerating, as opposed to what you see, and the promise of myostatin, right, has been that you're going to look like this, right? And so, okay. Now, what about follistatin? Well, Scott Harper's lab published a few years ago in their FSHD-like mouse model that if you treat them with follistatin, so even though, I just showed you, even though you have very low levels of myostatin, if you do a follistatin treatment, you can actually put on mass in the mouse models of FSHD. Okay, and this has actually been seen with other myostatin targeting pathways. So there is a window of, there is the potential in mice that are expressing DUX4 to put on a little bit more muscle mass. And here they actually showed a little bit gain in force and functionality in some ways. Okay, now, so far this hasn't translated to humans. Okay, so what's going on, right? So we talked about that. You have myostatin, follistatin. So then, I always tell you the devil's in the details, okay? So when it comes to blocking myostatin, right, there's different ways to do it. You can block the active form and the inactive form and bind that. And this is, I believe, where the Roche antibody was working. You also can block the receptor. And there have been clinical trials in FSHD where they block binding of the myostatin to its receptor. And also that should then stop signaling of myostatin. And but again, these clinical trials failed to show functional benefit in FSHD, okay? And it's not just FSHD. This is also Duchenne muscular dystrophy. In fact, Roche recently stopped their Duchenne program. Pfizer stopped their Duchenne program. Okay, so we always call it the promise of myostatin inhibition because you keep seeing bodybuilders, and wanting to do follistatin to bulk up. Animals can bulk up. But again, these are healthy individuals. When you have a dystrophy, there's been very limited success. And here's just some example. Monoclonal antibodies that directly bind myostatin, right? These trials have been discontinued. Receptor-based inhibitors. Remember the Acceleron trials? There was ACE83, ACE31. This is from Acceleron. These were in FSHD. These are blinding the receptors. These are one of the first FSHD clinical trial a number of years ago by Acceleron. And that, again, they showed increased muscle volume, but no increased strength. Small molecule inhibitors, follistatin therapies, these are still kind of going. There's CRISPR and RNAi approaches, okay? So the Roche trial was a monoclonal antibody targeting circulating myostatin. It's in trial for SMA when combined with an SMA targeted therapy, and in trial for FSHD as a standalone, okay? And both of those trials were stopped for the same reason, not consistently improving muscle growth and function, okay? Now, we told you not too long ago about Scholar Rock, okay? Now, Scholar Rock has a similar approach. So why is Scholar Rock, if they have a similar approach and Roche just failed, why are they still going forward? In fact, with all this whole laundry list of failures, why are they still going forward? Okay, well, as I mentioned, Roche halts development in SMA as well as FSHD. And the SMA trial was in conjunction with an SMA therapy. SMA already has 4 different FDA-approved therapies that restore SMN function and protein levels, okay? So why is Scholar Rock going forward? Because Scholar Rock actually did the same experiment. They actually used their myostatin inhibition antibody, a monoclonal antibody against the latent form of myostatin, and they combined it with an SMA therapy, and they found benefit, actually. Okay, went through phase 3, and I believe it's recently got FDA approval to be used. They actually saw benefit. Now, and so what do we see? They had succeeded in a phase 3 trial in SMA. Okay, I believe this is a little bit of an this is an old report. I believe they have FDA approval already, and it's called abitagramab. Okay, and so this already sets it apart. So devil's in the details. The Roche similar strategy didn't show any benefit. Scholar Rock, same strategy, did show clinical benefit. Now remember, SMA is a motor neuron disease, not a muscular dystrophy, but I believe this is the rationale for going forward into FSHD, that maybe they would have some better success than Roche. We will see. So what do they say? This is actually, it's called the phase 2 Forge trial. This is one of their posters. And I don't know if I can zoom in on some of this stuff so you guys can see it better. But basically, this is the idea. You have inactive, you have promyostatin, latent myostatin, and so it's actually functioning up here, binding up promyostatin and latent myostatin. This is where the antibody's taking it out of the system, okay? And presumably, you'll lead to an increase in protein synthesis and skeletal muscle growth, okay? Now, I gotta tell you, personally, I would much rather see this in FSHD combined with an anti-DUX4 therapeutic. That's where I think this could have some benefit. I'm not a fan of this as a standalone. I'm just telling you that right out. I wish them the best. I hope it works. But my excitement over Scholar Rock is as a combinational therapy similar to what they're seeing in SMA. But we'll see. All right, so that's the first bit of news. The second bit of news that came out, that EpiCRISPR, actually great news for EpiCRISPR, right? Gene therapy company in the space. They were named Fast Company's annual list of the world's most innovative companies of 2026. So congratulations to Amber Saltzman and the team, and Stanley Chi and everybody over at EpiCRISPR. I'm sure they don't really care what I say about them. They have the Epi321. This is an epigenetic gene therapy that's supposed to shut off DUX4 by methylating the D4Z4, and one-shot cure-all permanent fix for FSHD. I've detailed in great detail. I've expounded in great detail on my concerns about this trial. And I realize it's a phase 1/2. They're only looking at 10 people, and it's really about safety and tolerability, but they do have a whole lot of secondary outcome measures they're going to be looking at. And the thing about this, when this came out recently, is they actually pointed out that they have 8 participants have been dosed, and that they are already reporting favorable improvements across surrogate biomarkers and functional measures. Now, the only thing that's not a surprise is the no serious adverse events. So they caution this clinical data is preliminary. But they do believe that they're seeing muscle strength gain and increased speed time assessments and gains in muscle mass. Now, again, the devil's in the details, and there's a previous podcast where I discussed this. But now this is going to be compared to there's no placebo in this. Everybody in the trial is on the gene therapy, and they know they're on the gene therapy. And they're being compared to a natural history study where everybody is not on a therapy. So you're taking a bunch of people on a drug that are hoping to get better and comparing them with a bunch of people that know they are not on a drug and they know they are going to get worse. And they're saying that they see favorable improvements, which is not a surprise. Show us the data. Talk is cheap. Show us the data. Because you know what? You could have a favorable improvement of 0.1 seconds in your timed up and go. And you could have a favorable improvement of 2 seconds, which actually it's a very short essay, but could be a big difference. Are these actually important improvements? Anyway, I'm sure they will show us the data. I'm just impatient. I love to see data and interrogate it. And I'll tell you, if the data is good, solid data that truly shows an effect, I will be the first one to say, wow, congratulations, I was wrong. And I really will. You'll hear me say I was wrong when I am. You should be around our house more. You'd hear it a lot. All right, moving on from EpiCRISPR. And again, we really do want all clinical trials to succeed. And my concern with some of the companies I criticize is that either their data, in my opinion, isn't strong enough to warrant going to trial, or even if their technology looks promising enough, the trial design is not giving their therapeutic a fair shot at success in the trial. Actually, when it comes to EpiCRISPR, I think both of those things are true. I had concerns about their preclinical data, and I have tremendous concerns about why they're using AAVrh74. We're not going to get into that, but that doesn't mean we don't wish them the best, hope for their success, and we don't ever celebrate a failure, even though we predict it. I would celebrate being wrong and say no one could be happier about being wrong than having a trial that was a spectacular success. So we do appreciate everyone in the space, but I do hope I'm wrong on this, but we'll see. All right, switching gears a little bit. It's kind of but if you don't know who Frederick Sanger is, he's actually an incredibly remarkable person. He actually won a Nobel Prize in chemistry for protein sequencing. Show how to sequenced insulin, first protein sequenced, an amino acid sequence, how to do that. And then he just went on. He said, "Oh, screw. Okay, we got protein. Let's do DNA." He then figured out a method for DNA sequencing that's still used today, the dideoxy termination method, the Sanger sequencing. Actually, we still call that our very rapid turnover diagnostic. We actually use Sanger sequencing instead of next generation sequencing. It's still good today. But what I really admire about him is that at age, with all of this success and the top of the career and king of the world, very humble guy, actually just walked away from science at age 66, retired to spend time with his family, hobbies, boating, and gardening. And I always thought, boy, that's, you want to make your mark and then go on to your next chapter of life. Gotta tell you, I'm 57. I haven't done anything like this. But I always have him in mind when I think about, God, make your mark and walk away. And that may be a theme of our lab in the near future. Anyway, but the reason I bring it up is this great rule, Sanger's rule. Anytime you get technical development that's 2 to 3-fold or more efficient, accurate, cheaper, a whole range of experiments open up. And this has been something I've been trying to hammer home when it comes to the antisense, right? Avidity is an siRNA. Arrowhead, Sarepta is an siRNA. Myricule is an antisense. Dyne is antisense. It's all siRNA, antisense, whatever. The devil's in the details. And there can be all these different ways and avenues of being more efficient. They're not all the same. They're all targeting the DUX4 mRNA, but they're not all the same. And so this gets us to the last kind of news item, which was this data from Sarepta that came out. So Sarepta, which again acquired the FSHD program from Arrowhead. I believe Arrowhead still exists as an independent entity. But what caught my attention here was this. Sarepta's alpha-5 beta-6 integrin targeted siRNA approach achieves high muscle concentrations. Okay? Why did this catch my attention? I'd have to go back and listen to what I've said in the past. But avidity targets the transferrin receptor. And this actually causes some problems if you get too high a dose. You have a dose-limiting effect where you start getting adverse events. And this leads to a ceiling for muscle concentration in DUX4 knockdown. You can't just keep adding more. In fact, they saw this in their myotonic dystrophy trial. And so all their siRNAs have an antibody that targets the transferrin receptor. And I was thinking that that's what everybody in the space was using. Honestly, I thought that maybe Arrowhead was using that same antibody. I don't know if I said it. I'd have to go back and look. If I said it, I was wrong. I shouldn't have assumed that's what they were using. They may never have disclosed it. But when I saw this actually, boom, really lit up my senses because it is a different targeting approach. It's exactly what I've been talking about, which is now Souffle also has a very different targeting approach. They are not targeting the transferrin receptor. In fact, they won't disclose what they're targeting. But targeting is one of the key differences in these different antisense approaches going after DUX4. The devil's in the details. How you get it into cells is going to matter. Why is it going to matter? And this is actually spectacular news, right? So here we go. We are pleased that these early clinical results showed high levels of siRNA delivery to muscle with no saturation of muscle siRNA uptake or dose-limiting safety signals to date. We believe this supports the differentiated potential of this siRNA platform and strengthens our belief that this approach could meaningfully change the treatment landscape for patients. Okay? These preliminary clinical data show consistent dose-dependent increases in plasma and muscle drug exposures across clinical and non-clinical studies and suggest that alpha-5 beta 6 integrin targeting ligand mediates robust siRNA delivery. Okay? And they hypothesize this will ultimately enable higher dosing that translates into clinical efficiency for patients with FSHD and DM. That is spectacular news. In fact, that is actually why when I started this out, I said today, even though I haven't seen their DUX4 knockdown data, I haven't seen any of they haven't released that I'm aware of, any of their actual clinical data from their Arrowhead Sarepta trial. I've seen the preclinical data, very comparable to avidity. They have great preclinical data in our mouse model and in human muscle cells. The concern with avidity was that doubling the dose didn't give you any benefit and actually started to give some problems. So you kind of have a dose limiting. This is what you got with avidity's current technology. I'm sure they're working on different modifications. But with this technology, it appears to me, as a scientist in the space, that this is actually an improvement because you want to get as little DUX4 as possible. You want to knock down as much DUX4 as possible. You want to get as much safely of this siRNA into your muscle cells and also, hopefully, not into other tissues. So how does this work? Now, the one thing about alpha-5 beta-6 is it is expressed exclusively on endothelial cells, okay? So what's going on is, but this is actually how AAV gets into cells, so it kind of makes sense. This is actually so they're using the same mode of entry that gene therapy companies use to get their viral delivery of their DNA into the muscle cells, and they're using that for their siRNA. So basically, the integrin alpha-5 is a heterodimer with integrin beta-6, and they are heterodimer, and they're involved in TGF-beta signaling. But what happens is if you bind to this heterodimer with either your siRNA or this is what the viruses actually bind, there's something that's called transcytosis that happens. Transcytosis, okay? So they are internalized into the cell. So how does this happen? Well, I told you these are expressed on endothelial cells. all of your blood vessels are coated. The inside of your blood vessels are all endothelial cells. That's what it is. So you have endothelial cells that are kind of lining this, and it lines your organs and your blood vessels. It goes throughout all of your blood vessels. It's binding on these receptors in the blood vessels, and then it goes to this transcytosis, where it actually internalizes, transports across the endothelial cell, released via exocytosis on the muscle side, and you're able then to again internalize on surface receptors into the muscle and infect the muscle following endosomal escape and nuclear transport. And this is incredibly efficient. This is actually how AAV myotropic vectors get into muscle. Okay, so it's it's the same system, and so it's very different than the transferrin thing. It's a different molecule, and it appears to not have any, or at this step, any dose limitation. So it's just really cool technology. Again, the devil's in the details, right? It's an interesting technology. I gotta tell you, the viruses that we use in our lab takes the same mechanism. They're targeted to the same receptor to bring our gene therapy into the muscle cells. Hey, I'm back for a little bit of an update. So I gotta tell you, when I was looking at the Sarepta data, I got so excited over the alpha-5 beta-6 receptor targeting instead of transferrin. The transferrin receptor, right? I mean, I know there are all the receptors that are being targeted are going to be on endothelial cells. And this is how they get into muscle. They all get into muscle kind of on the same, in some degree, on the same way, but there are differences. And the thing about the transferrin receptor that Avidity is using and other companies are using is that they have them. You can basically have a ceiling. They get totally saturated. They don't recycle fast enough, and that you actually can get adverse events when you go higher. That's at least what Avidity showed. So one of the reasons I like Souffle so much, one, they have better durability in their preclinical data that they release, but also they have a different type of muscle-specific targeting, muscle targeting. They're not using the transferrin receptor. I actually wasn't really sure what Sarepta was using. It turns out, so they're using the alpha-5 beta-6 receptor, integrin. When I'm so excited, and I still am. I'm just like, yay, somebody's using a different receptor. It's published. And my prediction was that this would be much better than the transferrin receptor. And that is why I said my new number one is going to be Arrowhead, because my prediction was, and honestly, at the time I hadn't seen the data my prediction was that if you had this better targeting, it would work better. So in some ways you can say blind prediction. I'll put a positive spin on me scrolling too fast and missing the data. So there we go. I should have spent more time, but I do have a day job and we are a bit overwhelmed in the lab as we tend to be. So here, let's go to the data really quick. So I'll share my screen right here, and so and thank you for pointing it out. My listeners pointed it out. They said, Peter, you said you haven't seen the data. The data's there. And so yes, that's why it's just like wake up Sunday morning and wonderful Sunday morning to say, Jones, you're an idiot. I know that. You guys know I'm an idiot. Anyway, so here it is. An N of 6. Now these are looking at these are treatment cohorts pooled from 1 mg per kg and 2 mg per kg, and cohort 3 is 4 mg per kg. Okay, so they pool these together. This is the data, and they're able to show basically 90% reduction of DUX4 target genes. As you know, DUX4 is a transcription factor, activates target genes. So instead of assaying for DUX4, you assay DUX4 target genes. There's kind of several traditional pools. There's the pool of 4 target genes here, the pool of 6 target genes here. There's some overlap, which makes your pool of 8 target genes here. And what they show is it doesn't matter which set of target genes you look at, you're going to get greater than 90% on average. And it looks like the low end is actually going to be pretty darn good. So now the devil's in the details. You have your placebo control. You can see there's wide variability in the levels of target genes beforehand. It would be nice if they broke this out on the individual basis. That's actually what did, and see the individual 1 went from here to here, individual 2 from here to here. It'd be nice to know if everybody went down or what, but on average, showing 90%, even though it's an N of 6, that's fantastic. That is great. That is amazing. Actually, some people showed 100% reduction in their target gene versus their baseline, and that's amazing. So it's exact. So in some ways, I'm an idiot. I missed the data. I'm happy to admit when I'm wrong or when I miss something, and we want to correct it. But on the other hand, it blindly predicted that by getting away from the transferrin receptor and into the alpha and into a different receptor, in this case, the alpha 5 beta 6 integrin receptor, it's actually going to be better. And it is. Then they went through and they did serum creatine kinase. So these are the two data slides. I don't know why I stopped just before this. I was so focused because it doesn't matter. It doesn't matter. I don't know why I stopped before. I assumed this was the myotonic data. Anyway, if you look at the creatine kinase, so what is creatine kinase? This is an enzyme that's released from your muscle cells when they degrade. And so in Duchenne muscular dystrophy, which has leaky membrane due to lack of dystrophin, you have very high levels in the thousands of serum creatine kinase. In FSHD, your levels can be like 300 to 800, whatever, higher than normal. When you exercise, the levels go up. That's why it's not a great marker. Exercise at high activity. So it's much more variable than the KHD-CL1, which is what Avidity is using as a serum biomarker. But Avidity has proprietary rights on that and intellectual property, so other companies aren't allowed to use it at this time. But it turns out that, KHD-CL1 tracks about the same as creatine kinase in the Avidity trial. Some company, so they're not a whole lot different. I think the main difference is going to be the fact that CK levels can change, again, due to activity and give you a false result, a false high when maybe it should be down. But regardless, I'm looking at this is the 4 mg per kg and the 8 mg per kg treatment pool. And again, they're looking here, well, they have N of 31, right? Following a single dose administration. Now you can assess serum much better than you can assess biopsy data. This data here has to come from biopsies, right? And it's going to be looking at qPCR, so looking at DUX4 target genes from muscle biopsies. Here, serum creatine kinase, you're just looking at in blood. So it's much quicker and better to do. And what they do show is that you get a very rapid reduction that is maintained over time in the 33% here. You'd still like to see a little bit better, but actually it's all trending really good. So there you go. So that's the up. Now I've actually seen, I was talking with Brad, and Brad was like, why didn't they release that? I don't know. They must be releasing. I said, well, they must going to have another release. It was there. It was my mistake. Again, I'll be the first one to own up anytime I make a mistake and anytime I overlook something and anytime I'm wrong, I want to correct it because we want to give you the best information. You know what? I'm doing my best. But I apologize for missing that. And thank you so much for keeping me on my toes and letting me give me the opportunity to update it. And also for Sarepta, I apologize for missing it. But remember, based on just the differential targeting and the excitement over how it's going to target, it's better than I expected, right? Predicted it would be good, and it is. So maybe I'm not a total idiot. Well, that's up for debate. Okay, so that was the first thing I wanted to cover about the previous podcast, talking about I want to change my vote. Say Arrowhead is what I would choose if I had to choose right now. Now, it's an interesting thing because I looked and I don't think there's any place where you can choose between Avidity and Arrowhead because none of them, if I saw it correctly, I just was on clinicaltrials.gov, are in the same sites. So you don't actually really need to make a choice. But just conceptually, based on the science, I think right now I would have Arrowhead, number one. So the other thing Brad had asked, someone had asked a question on the Reddit feed about, there was some question about if you're in a phase 1/2, do you go in and become part of the phase 3? And I said, well, no, the phase 3 is a completely separate entity unto itself. Well, it is as it isn't, because it turns out, as I was looking through the ARO-DUX4 trial, which is now the Sarepta 101 trial, same thing, they talk about that there's different parts of the study, right? And so participants who complete part 1 will have the option to rescreen and re-randomize into part 2. And participants will undergo pre and post-dose MRI guided muscle biopsies, 2 biopsies. So it sounds like then you'd get multiple. And participants who complete part 1 and enroll in part 2, they do have to have an additional biopsy. However, you will have the option of continuing to receive drug in an open label extension study, or you can then go into the phase 3 study, is what it actually says. So it sounds like all the options are on the table. If you are in the phase 1/2 trial, based on what I'm reading right here, it sounds like you would have the option to go into the phase 3. Now, it just seems like it's a whole different kind of arm, and I guess they will keep track of it. I don't know. I'm not a clinical trials expert by any means on how they design these. I was just quite surprised to find this, and I want to, of course, always want to make sure that I get it correct. When you go through the consenting process, if you're going to talk with these companies, you'll be able to ask all these questions and find out what are your options at the end of the study. And we're going to talk about that in a second. But here, open label extension. Conceptually, if I was in the phase 1/2 and they said, well, you can go into the phase 3 and start again, or you can just have open label extension of what you're on, it just seems like, why wouldn't you do the open label extension unless there's a dosing issue that maybe you, anyway. Which brings us to the next thing I wanted to talk about. So I made a comment on the previous podcast that somebody who was in the avidity trial commented that they're going to have the drug for the rest of their life because they're in the trial. And I said, well, maybe they are, maybe they aren't. I don't actually know. You better look into it. Okay. And somebody commented to me that, Joan, don't you know what you're talking about? You're always given the drug for life if you're in a clinical trial. And I said, well, I don't think that that's true. So what are you going to do? What do we do? We're scientists. Let's go right to the resource. So this is all in the World Medical Association Declaration of Helsinki, Ethical Principles for Medical Research Involving Human Participants. Okay, and now we're going to get out of this and we're going to go to the web and actually open up the site and see what's going on. And so we're going to go and see, here is the Helsinki Declaration, and you can find out what countries have agreed to in regards to human research, right? General principles, it binds the physician with the words, the health and well-being of my patient will be my first consideration. I hope that is true. You sure do hope that is true. The physician must commit to the primacy of patient health and well-being and must offer care in the patient's best interest. And I take this to mean that an MD then is declaring they would never promote a clinical trial that they themselves did not believe in the science, right? I think that's how I interpret that, is that if you don't believe in the science and you don't believe that this is a good clinical trial, then you should not be promoting it. And I know that there have been some clinical trial sites that have refused some clinical trials because they didn't think it was very good, and I have tremendous respect for those individuals. But I guess it's on the individual to decide. I guess there's no right or wrong answer. It's how you interpret the data. Okay, so let's see, what did I want to get to? You can read through here. It's kind of an interesting read. I'm not going to go through the whole thing, but there's risk, burdens, and benefits. Here we go. Physicians and other researchers may not engage in research involving human participants unless they are confident that the risks and burdens have been adequately assessed and can be satisfactorily managed. When the risks and burdens are found to outweigh the potential benefits, or when there is conclusive proof of definitive outcomes, physicians and other researchers must assess whether to continue, modify, or immediately stop the research. Okay, so this might get to what Roche was talking about, right? It was safe, but the data suggested that there was not any real benefit to continuing the trial. Under the Helsinki Declaration, right, Declaration of Helsinki, whatever, right? It is appropriate to stop a study that is not showing benefit, right? Now you can decide what is conclusive and what's not. The Fulcrum trial was not showing benefit either, but that was debated. Everything is debatable. The people running Fulcrum, they felt there was enough indication of benefit. I did not. The people running Roche do not feel there was enough benefit, so they terminated it. You got to respect both decisions, because you don't want to stop a trial of something that could be a success, but you don't want to continue a trial and keep subjecting patients to experimentation when you know it's going to be a failure. At least that's my opinion. Okay, scientific requirements and research protocols. Okay, medical research involving human participants must have a scientifically sound and rigorous design and execution that are likely to produce reliable, valid, and valuable knowledge and avoid research waste. The research must conform to generally accepted scientific principles, be based on a thorough knowledge of the scientific literature, other relevant sources of information, and adequate laboratory and, as appropriate, animal experimentation. The welfare of animals used for research must be respected. Okay, so again, this is open to interpretation. I don't think anyone would disagree with that, and everybody going to clinical trial would say that they are adhering to that. And this is where scientists can disagree. The way we do our research, we have a different level where we need to say what we consider rigorous design and execution, and what we believe based on prior clinical data from others that have gone through clinical trials and research data in the lab. Everybody, I guess, has their own threshold of where they are. Boy, I just really gotta make sure that I don't have too much stuff here on my my screen that you guys are able to see. Okay. All right, so what about going? There's ethics committees, privacy and confidentiality, free and informed consent. You can read about this. It's actually not that long of a read. It's kind of worth checking out. Use of placebo. The benefits, risk, burdens, and effectiveness of a new intervention must be tested against those of the best proven interventions, except in the following circumstances. If no proven intervention exists, the use of placebo or no intervention is acceptable. Okay, right now there is no accepted treatment for FSHD, no accepted method for knocking down DUX4, therefore placebo is the best control. Now, and that is what Avidity is using. That is what Arrowhead Sarepta is using. EpiCRISPR does not have a placebo control, but they have the no intervention. They're actually comparing against a natural history group. Okay, that is kind of their no intervention group that they're comparing against. Okay, so if now one of the issues about an FSHD therapy getting FDA approval is, do you have to then compare your intervention against the FDA approved one? So let's say Avidity gets approved. Is everyone going to have to compare theirs to Avidity? And their answer is yes and no, right? Because it is acceptable not to do that if, for compelling and scientifically sound methodological reasons, the use of any intervention other than the best proven one, the use of placebo or no intervention is necessary to determine the efficacy or safety of an intervention. Okay, and the participants who receive any intervention other than the best proven ones, placebo or no intervention, will not be subject to additional risks or serious or irreversible harm as a result of not receiving the best proven intervention. Extreme care must be taken to avoid abuse of this option. So what it's basically saying is, if everybody in the control group is on Avidity's antisense, how are we supposed to assess if a gene therapy that knocks down DUX4 is actually working when or how well it works if you already have this background of a different type of DUX4 knockdown, an RNA knockdown versus a CRISPR therapy. So you can make scientific reasons, methodological reasons why you need a group that is not on an approved therapy, for example. So it's possible, but it's also possible that you're going to need to compare to that group. Post-trial provisions. Now, this is actually what I wanted to talk about. It took a long time to get to, right? Because the question came out, if you're in a clinical trial, do you get to have then at the end of the trial to be on the drug for free for the rest of your life? Now, if it's a gene therapy trial like EpiCRISPR, it's a one-shot done deal. Everybody, you don't get a second shot. It doesn't matter. Everybody who's on the trial is on it. Everybody in the trial is on the drug. In a phase 3, if it were to go to phase 3, there's probably going to be a crossover where 50% get the gene therapy, 50% get the placebo. Halfway through, you switch, and the 50% that got the placebo now get the gene therapy. At the end of the trial, 100% will be on the gene therapy, and you don't have any open label extension. Okay, it's a done deal. That's generally how a gene therapy is going to work. Now, something like an siRNA, like Avidity or Arrowhead or Dyne or Myricule, the question would be, well, at the end of the trial, where you've been on this antisense, you have an open label, are you ever going to lose access to that? And my answer was, you better check. You better check that there's an open label extension. You better check what the plans are for the future. And I was told, you always get that. I always say watch out for saying always. Okay, so what does the Helsinki Declaration say? In advance of a clinical trial, post-trial provisions must be arranged by sponsors and researchers to be provided by themselves, healthcare systems, or governments if all participants who still need an intervention identified as beneficial and reasonably safe in the trial. Sentence one, everybody in the trial is going to get a beneficial therapy for life. But the devil's in the details, because sentence two, exceptions to this requirement must be approved by a research ethics committee. Specific information about post-trial provisions must be disclosed to participants as part of informed consent. So what this says is most of the time, you're going to have access to a beneficial therapy, and it needs to be arranged, part of the clinical trial, but there are exceptions. And that's all I'm saying. I'm saying, you know, I hope in FSH, I hope there's never an exception. But you know what? We're going to get on the clinicaltrial.gov site, and we're going to look at things, and we're going to actually check this out. But these are things that should all be covered in the informed consent. And if it's not covered, you should ask, what happens at the end of the trial? Make sure there's an open label. Now, if there's not an open label, at least you'll be aware. And what happens, you know, if the drug's safe and benefit is going to be approved, are you going to have access to it forever? Just make sure. That's all I'm saying. Okay, I told you, I'm the dumbest person in the room. I just want to make sure. I'm not going to speak for any of these companies and have someone say, well, we were told that you, I said, I'm just saying right here, it says there are the possibility of exceptions. So check it out. That's all. But don't worry, I'm still an idiot. All right, last thing I want to do. Let's go to clinicaltrials.gov. All right, for those of you that are able to watch, I'll try to describe it good when I'm on. Okay, clinicaltrials.gov, condition, disease, FSHD. Type that in. What do we get? Okay, we get a whole bunch of search results. So what are we going to look for? I'm going to say recruiting and not yet recruiting. So there are 26 trials recruiting. There are 4 trials registered that are not yet recruiting. I'm going to go down here and look for, what am I going to, I want to say interventional trials. Your choices are interventional, observational, like patient registries, expanded access, things like that, of which there are none. So you really in FSHD, your choices are interventional, 59, or observational, 36. Apply filters. All right, so what do we get here? We got 17. Okay. Which is interesting because 4 are in phase 1, 3 are in phase 2, 1 is in phase 3, none are in phase 4, and 11 are not applicable. Okay, so what are they? Testing a tailored home exercise program to reduce pain and fatigue in patients with FSHD. Sounds interesting. Not much information on it. Here we go. Safety and preliminary efficacy of ULSC. So this is the umbilical cord derived stem cells and FSHD. This is at Stanford. Let's punch this one up. That's that company Resteem LLC. This is basically, they have some umbilical cord derived stem cell line that, as far as I'm concerned, is just going to be pumping out immunosuppression and some growth hormones locally wherever it's injected. This is being done at Stanford, and people have been asking for my opinion. You know, it's safe. Are you going to get some benefit? It's, you know, we do believe, you know, there's an inflammation component of FSHD. Things that decrease inflammation are likely to be beneficial. You know, Baja Blast, man, jump in the cold plunge, you know. But personally, this is not for me. This is not one that I would personally sign up for. You know, but that's me. It just, again, it's immunosuppression. But again, we'll see. I'm glad the trial is going on because there's a lot of questions about, do you get any benefit? And so while I'm happy someone else is doing the trial. If it were me, I would not sign up for that trial. Study of AeroDux4 in adult and adolescent patients with facioscapulohumeral muscular dystrophy type 1. 17 locations. Okay, let's check out these locations. This is New South Wales, Australia, Canada, Germany, Italy, Netherlands, New Zealand, Spain, and they withdrew the Thailand. So pissed they withdrew the Thailand site. I got to talk to somebody over there. I want to get my friend T down to Thailand, and this is the trial I would want to get her into. I like this. I'll tell you right now, as of today, this would be the #1 trial I think that I would be interested in signing up for if I were an FSHD patient. But you know what? I'm in Carson City, Nevada, and I don't have access to this. So that being said, that's what's going on. Okay, phase 2 study evaluating bicrogromad for FSHD. This is the Scholar Rock study. Okay, this is going to be myostatin inhibition with an antibody. It is a randomized, double-blind, placebo-controlled 52-week study. Okay, I got to tell you, I showed you before, it showed benefit in SMA when combined with an SMA therapy. The history of myostatin inhibition in FSHD as a standalone treatment is horrible. Boy, I gotta tell you, I would if this were combined with, say, Avidity or Arrowhead or Myrcure, any of these other ones, I'd sign me up. But as a standalone, I'm not too high on this one personally. But I hope people sign up for it because I'd sure like to see. It does show it is different than the Roche drug. The SMA trial shows that it has more potential benefit. So what's the next one? Okay, first in human study, Epi 321 for FSHD. This is EpiCRISPR. I think I've been, it sounds like they've got 8 out of their 10 people already injected. I think we're looking for 10. This is a gene therapy to shut down DUX4. I've well documented my reasons why I personally would not sign up for this one, but I wish them the best success. Study to evaluate Delbrax, AOC 1020 in participants with FSHD. This is the Avidity trial. This is in the U.S., 22 locations, California, Colorado, Florida, Iowa, Kansas, Maryland, Massachusetts, New York, North Carolina, Ohio, Pennsylvania, Texas, Virginia, Canada, and Japan, 4 sites in Japan. This is their phase 3 trial. You know what? This isn't a bad second choice. Actually, considering where it is and there's no overlap in the locations with Arrowhead, if I was not in one of those locations. Now this gets to this, I would sign up for Avidity. I still stand by that. Now, it never occurred to me until a couple days later why someone was asking me this. And then combined with what we just talked about, you get to be on the drug. Do you get, assuming that if you're in one of these trials and it shows benefit, you will then be provided this drug for life so that you always get to have your necessary treatment. Because as soon as you go off the drug, the DUX4 is not getting turned off. You have to be on these antisenses for the rest of your life. As soon as you stop taking antisense, the DUX4 mRNA will not be degraded and protein will be made and pathology will start again. So you need these the rest of your life. So if being in this trial and rightfully so, that you're going to get these for the rest of your life, then maybe it does make a difference which trial you try to get in. But this is the caveat. You don't get to choose necessarily. You can choose which trial you make yourself available to, but they might not choose you. Second, they have different locations around the world. Avidity needs you every 6 weeks. Can you go to, can you go to where they're needed? If you're in Europe, can you go to a site that has Avidity every 6 weeks? If you're in the US, can you go to an Arrowhead site? Now I don't actually know how long Arrowhead is a longer time frame between injections, and that's a phase 1/2. When they do their phase 3, they'll have a phase 3 will be more of and it might actually be phase 3. I hope phase 3 is also in the US. And come on, let's get Bangkok back on board, or somebody in Asia. Let's get somewhere in Asia onto that Arrowhead trial. Come on, guys, and there you go. So it's So it might make a difference. I don't know if that's what people are thinking when they're asking the question. Are they thinking long term? And again, this is where I'm not a patient, so I'm just thinking strictly of the science. So when I really cannot get into your head as a patient. All I can say is scientifically, based on everything that I know, I would say right now that when I look through all of this, there's one more. There's clenbuterol to target DUX4 and FSHD. I'm actually part of the Wellstone Center that is sponsoring this, so that is why I have not talked about it. I think it's a conflict of interest, and I should not discuss this trial at at all. Check it out, look online, and contact Rebecca Clay at Kansas University Medical Center and you can find out more about it if you're interested. But again, I'm actually part of the Wellstone. This is part of our Wellstone grant, and so I'm not going to say anything beyond that. And I'm going to call a stop to that and come back here. All right. So the clinical trial summary that I would basically say, looking at everything, considering trials that you could sign up for now that are basically on the board up in clinicaltrials.gov, from my perspective as a scientist on what I know about the science, I'll say my number one that I would go right now, without having seen data from the clinic yet, other than safety data, the Arrowhead Sarepta trial, the Arrow DUX4 or whatever it was called, the Sarepta 1001 is the same thing, mainly because I like their antibody targeted delivery as allowing for a higher dose, more effective muscle uptake, and just basically better delivery, which I believe could come into better efficacy. Number two would be Avidity that's out there. We've seen their preliminary data, haven't seen their extended data. Looks like it's working, looked like it was working in everybody. At least you're going to get some benefit between 25 and 80% benefit on an individual basis. Hopefully it's even more. What I don't like about Avidity is they have a dose limiting kind of toxicity with targeting the transferrin receptor. They're not getting so that means that limits how much uptake they can get and how much knockdown, and the durability seems kind of suspect. Every 6 weeks is a lot to do. Now, I don't know if you'll have to do that the rest of your life. Maybe you'll build up at some level and you can extend it out, but that just seems, I would rather conceptually thinking it would be nicer if that was longer, but that's the kind of like the clubhouse leader because they're the only ones that have shown something's working. Beyond those are right now the only two that I would actually, again, taking clenbuterol off the table for me because I have a conflict of interest with that one. Those are the two I would sign up for. Not a fan of the umbilical cord stem cells. I'm just very cautious about whether that's going to provide any benefit. Skeletal, I think, is the most hopeful myostatin inhibition technology that I've seen, but considering the history of myostatin inhibition for FSHD as a standalone, I am very cautiously optimistic. As a combined therapy with a DUX4 knockdown, I would be much more like I think that would be fantastic. I hope that's where they're going. And what else? So that's kind of like what's out there on the space. So hopefully Dine and who's been talking forever about going to clinic, Soufflé. I'm a real big fan of the preclinical data that Soufflé has and their durability and their muscle targeting. They have said that they're going to have a clinical trial. Again, talk is cheap. But hopefully they'll have a clinical trial in 2026. I would be very positive about joining that clinical trial, as I would with Myracule. When Myracule gets their act together and goes to clinic, everything I've seen from them is very favorable. I don't know who else. I guess there's some others. I got to be careful what I say and what I disclose. I know, but there's some good stuff coming. But thankfully, there's some good stuff already here. At least showing some benefit, and we are thankful for everybody that's in the FSHD space. Last thing I want to do is a little clarify. I always catch crap for this. One of the things I do in the podcast, I just try to tell you what it's like to be an academic in the space. All I can tell you is my own personal experience. I don't know anyone else's experience. I just have it from my perspective. And sometimes it comes across, as people point out to me and put me in my place, that, hey, Jones, you're not the only one making sacrifices in the FSHD space. I know that. Everyone knows that. So just to be clear, and I will say it, pretty much, I understand that everybody who works for one of the FSHD foundations could likely get a higher paying job working for somebody else and are making a sacrifice, a financial sacrifice and a personal sacrifice for the benefit of the FSHD field. I understand that. I understand that many researchers that are academics treat their labs, as we do, as a small business. And you make sacrifices of time, family time, personal time, financial sacrifices, just as we do. And so to keep the business running. Times are tough, funding's tough. You got to keep going. You support your students. The last person that gets paid is always the boss. I tell you our experience. When I tell you my experience and how we run our lab, I do not mean that to say that we are saying, gee, we're better than everybody and we're the only people making sacrifices. What I was actually trying to say is we're just part of the FSHD community where everybody is making sacrifices, almost everybody, for the sake of the field. Patients makes, you guys that join clinical trials, it's a tremendous sacrifice. If you're a caregiver, it's a tremendous sacrifice. Patients, you're staying in the workforce. Everybody in the rare disease space, that's just part of it. We all make ours, and all we're saying is we're doing our part. That's all. The frustration that comes, I'll just give you an example of how we're doing our part. The frustration comes up, and maybe it didn't come across, is that when you're killing yourself in the lab, you're doing everything you can to keep open, and then your boss, like at the university, says, oh, you're just a lazy slug. Really? You're not even around to know whether we're a lazy slug. That's frustrating. I hope all of you can appreciate that whatever your job is, that if you really just knock yourself out, you work really hard and you really do a bang up job and you show it to your boss and your boss is like, you don't feel great, do you? Well, that's how we are. All right, Brad's going to be back. We're going to be doing part 2 of the Reddit questions coming up this weekend, but I thought this was a good chance to interject. I thought I had a little time window there when Takako was down in the mouse room, but she caught me. I'm supposed to be working on our paper where I gotta tell you, last thing, just between us. I'm putting together the data on our 4,000 samples from our MyFSHD saliva testing, the at-home stuff. It is unbelievable. I mean, we got the coolest. I think this is going to be a unique data set that honestly, I don't think anybody in any rare disease has a data set like this because nobody does what we're doing. I got to tell you, I'll tell you one last thing. I had a call the other day with a medical doctor who thinks he's got FSHD and has FSHD in his family. And he was just like, Peter, you got to get some help. First off, he couldn't believe that I answered the phone. He's like, never even occurred to me I'd be talking to you. I figured I'd be talking to your assistant. I don't have an assistant. And he said, well, I sent my sample in. I'm like, oh yeah, I remember. I had the super extra bubble wrap because your tube got crushed last time. He's like, yeah, how do you know that? Well, I'm the guy that unwrapped the... He's like, Peter, you got to get some help. And I got to say, it was actually refreshing. It was very nice. But what's really nice is what's coming out of this effort, our little red hen effort, where we had to bake the bread ourselves. It's one hell of a loaf, let me tell you. And all these people that wouldn't help us, boy, you're going to wish you were part of this, man. You're going to wish you were part of this. Anyway, some people did help us, and I always want to finish here on a high note. I really will finish here on a high note. Big shout out to Friends of FSH Research, Terry Colella, Rick Colella, George Shaw, Bradley Bagshaw. They have supported us with our MyFSHD saliva testing, and they have supported us with the pig. And so I always want to give a shout out to those guys. Oh, and Neil Camarda, FSHD Canada, #1 supporter, supporting the pigs, supporting muscle building projects, supporting all sorts of cool technology. And my man Chip Wilson, independent of Solve, Chip on his own through his family foundation, funding stuff that Solve isn't smart enough to fund. Thank God for Chip and his support. He's smart enough to know what needs to be funded. Too bad his foundation isn't. So really want to end with a thanks to those three. Great supporters, wonderful supporters of the lab. We couldn't do it without you. Please, I got to make sure you know that we do truly appreciate you. All right, man, out of here.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. Do you have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information